Noswaith dda a chroeso I bawb. Good evening and welcome everyone. Thanks for joining us this evening. My name is Colin Reardon. I'm the Vice Chancellor of Cardiff University and I'll be hosting the event this evening, which, as you'll have just heard, is being recorded. And we will send you the recording link after the event and you'll be welcome to share that. So if you want to catch up on it at more leisure. So this evening we're showcasing research which helps us to understand how the body's own defense systems, the immune system, has a role in the way that dementia develops. And talking also about how we can hope to slow down, reverse or even prevent the onset and symptoms of the devastating disease of dementia, which takes a number of forms, of course. I mean, this is something that affects almost all of us one way or another, if not directly, then through our family and friends. So it's gratifying to see the large numbers who've registered for this event, but sadly not surprising. I'm delighted to be able to introduce our speakers today, Dr Mat Clement and Dr Wiola Zelek. They're research fellows from the School of Medicine, and they're also alumni because they both got their PhDs from us here at Cardiff. Before we go on to their talks, just a word about the context of their research in the environment that we have here at Cardiff. We are very much committed to nurturing the next generation of talent to take us forward in terms of tomorrow's breakthroughs in research, because research really is a pipeline. You have to start with students, moving through to research fellows and professors, and you need that kind of pipeline of talent, and Matt and Wiola are a really important part of that. Now, mind, brain and neuroscience is a research priority area here at Cardiff University, and because of that we've invested in world-leading research infrastructure, and it really is world-leading infrastructure. There's only 2 or 3 places in the world where you would find the combination of facilities that we have here at Cardiff. We've set up the Neuroscience and Mental Health Research Institute. We built Kubrick, which is the Cardiff University Brain Research Imaging Centre, which can produce images of the brain structure, the living brain, in really exquisite resolution, using very advanced MRI scanners and other scanners. And we also have a Centre for Neuropsychiatric Genetics and Genomics, and that investigates a range of neurodevelopmental and neurodegenerative disorders. Our researchers then are using these institutes and these resources to investigate the complex interaction between our genes, our cells, our brain tissues, and their role in maintaining cognitive function across our lives. So as a university, we want to provide the right kind of research environment that will support and nurture our researchers. And we do that in many ways, and certainly through the generosity of philanthropic donations. We support our early career researchers with funding for them to undertake work that provides leverage towards securing prestigious grants over time that will allow them then to conduct larger scale programs that in turn will deliver the discoveries we need to treat and one day beat neurological diseases like dementia. Now, after Matt and Wiola's presentation, we'll have time for discussion. You will be able to submit questions. We've received some questions in advance, so after their talks, we'll make a start with those. But if you do have questions, please put them in the Q&A box at the bottom of your screen. You can do that at any time, and we will answer as many as time permits. So please do take advantage of that opportunity. But for now, it's a great pleasure to ask Matt and Wiola to present. Welcome to you both. Croeso.
So hello, I hope you can all see my slides and hear me. So on behalf of both Wiola and myself, I'd like to thank the organisers for inviting us to talk to you today about the role of the immune system in dementia. In particular, what Wiola and I are interested in is the study of Alzheimer's disease, which is a form of dementia. So Alzheimer's disease is, as I said, a typical form of the most common form of dementia. It is basically a set of collective tasks, a set of collective symptoms that affects one's ability to perform cognitive tasks. So it's a very progressive disease where there's very early symptoms where you see loss of memory, loss of language, loss of orientation and concentration. And then it's later and more severely where the disease will develop into the loss of hallucination. You'd have more hallucinations and you'd have loss of body and motor control. And sadly, this is the leading cause of death in the UK of women, and also the second cause of death in men, even in the time of COVID. And this has a significant both economic and patient burden. And there are 850,000 people diagnosed in the UK currently with Alzheimer's disease. This is predicted to be 1 million people by 2025 and 2 million people by 2050. And it's not just a disease of the elderly, because there are approximately 45,000 people under 65 in the UK currently with Alzheimer's disease. But sadly, there is no appropriate therapy for Alzheimer's disease, and it is a very debilitating disease. And so that mine and Viola's research is undertaking novel immunological processes to identify new ways to treat this disease. But as I said, it has a significant financial burden, and Alzheimer's disease alone costs the NHS £26 billion a year. So this is approximately a fifth of NHS England's annual budget. So you can see the problems that you're dealing with. So how do you treat Alzheimer's disease and other forms of dementia? Well, it ultimately starts with how you diagnose. If you look at cognition here and the cognition versus disease duration, ultimately we want to be here. We want to be fully cognitive. However, as the disease progresses and tracks down this line, you will have a full-blown dementia, and as time has gone on, any form of therapeutic intervention is going to be pretty much useless here. So if you want to design effective therapies, you need to be initiating these very early on through disease. Because what Alzheimer's does is Alzheimer's neuropathologically is associated by the presence of these amyloid beta plaques and also these neurofibrillary tangles. These are deposition of proteins that exist in the brain that in normal people and normal non-Alzheimer's disease patients are perfectly cleared. So this leads to neuronal loss and degeneration, and the neurons, if you like, are those that send the signals to our brain and communicates to send the appropriate path. And then what this does is this leads to activated microglia and these cells called astrocytes. So these are brain-resident cells that live in the brain, and these become activated, and this shouldn't be happening. And this is what causes the disease. So that when we look at scanning using high-resolution scanning imaging, you can see in Alzheimer's disease patients, you see this upregulation of this protein called tau here, and then you can also see the deposition of these amyloid beta plaques here. So these things are naturally broken down in Alzheimer's disease. They form these insoluble plaques that can't be removed and causing the neuronal destruction. And sadly, this is what the brain looks like in a patient who has Alzheimer's disease. When you compare it to the brain on the left and the normal brain, you can see a complete loss of the global architecture and the resolute structure and function of the brain. And this is a cross-section of the brain here. You can see how fundamentally altered it is during the disease. So as a viral immunologist by trade, I was very interested to ask the question, can viral infections and the appropriate immune response that encompasses the viral infection actually contribute to Alzheimer's disease prognosis? So why is this important? Why? Well, because viral infections in the brain are very common. And in particular, I'm interested in a virus called cytomegalovirus. So this is a very ubiquitous beta herpes virus. This infects between 50 to 70% of the world's population, and typically is asymptomatic in healthy individuals. However, an infection with this virus will lead to morbidity and mortality in those that are immunocompromised. So these are AIDS patients, post-transplant recipient patients, and also in neonates, where congenital CMV infection is the leading cause of babies born with blindness, deafness, and mental disability. It's also important to therefore realize that as one ages, one also becomes more immune compromised. And the other thing to bear in mind with this virus is that even in high virus shedding in immunocompetent children, this is associated with an impairment of our overall T-cell immunity, and T-cells are the killer-like T-cells of our immune system that's involved with pathogen removal. So I wanted to ask the question, could cytomegalovirus herpes virus infection contribute to AD pathology? And interestingly, yes, it could. If you look at this graph here, we have what we call a discrimination ratio. Anything below 0.5 indicates that an individual is cognitively impaired. Anything above 0.5 tells you that an individual is cognitively intact. And if we look in red here, this is Alzheimer's disease individual with cytomegalovirus infection. Every single one of these individuals is cognitively impaired. But what's really interesting is that when we actually target the virus using antiviral compounds, we can restore the cognitive ability of these Alzheimer's disease patients to those similar to healthy controls. So, as I said, I'm a viral immunologist. Well, I was really interested to see what would actually happen to the brain tissue itself. And what I was able to do is I was able to enumerate the amount of viral specific T cells that are living in the brain. So in a healthy control with viral infection, there are some there. However, upon Alzheimer's disease with viral infection, the amount of viral specific T cells are tripled in the context of the disease. So this shows that the viral infection could be contributing to this cognitive decline in association with the immune system. So I was very interested, and Wiola will talk about this as well, as what impact this presence of viral specific immune cells has on the overall brain function itself. And the way we do this is using a process called dialytics. So this uses this advanced confocal microscope to actually look at, this is a cartoon, to actually look at the neurons. The neurons are the cells which communicate with one another, sending the signal through our brains. And what we're able to do is we're able to analyze what we call dendronic spines. These are the way, if you like, neuron fingers, the way they communicate with one another. And again, this is what the cartoon will look like, but this is what it looks like in real life. You can see the neuronal spines, the arms, the fingers of them communicating to one another. And interestingly, what we're able to do is we're able to look at the morphological change and the shift in the overall spine structure, and these are indicative of overall function. So I applied this in the context of the viral infection. What I was able to show is that you get reduced brain function of the neuronal cells in the presence of the virus. You can see all the spines here across all of the appropriate conditions. If you look here with the Alzheimer's disease individual with the virus, these spines are much more, they've shrunk down, not as widespread, they're not as fluorescent. So when we look at this overall, and we look at the spine density, what we're showing here is in the Alzheimer's disease with viral infection, we see a significant reduction in overall neuronal spine density, showing us that the neurons are not as functional because of the viral infection. So I've said to you that, that we have a difference in the neuronal function here, and we've also seen the presence of viral-specific immune cells into the brain. So what damage are those T cells actually causing to the parts of the brain that's responsible for our cognitive control? And so I took an advanced sequencing approach to ask that question. And because I'm interested in this region of the brain here called the hippocampus, because this region here is responsible for all of our memory and cognitive control. And the sequencing analysis enabled me to identify genes that are different that are being influenced by the viral infection, because this may identify new therapeutic markers of disease. So the way you do this is you take the brain, you generate coronal sections using these matrices, and this is what the brain ultimately looks like. And then I take microdissections of the hippocampus. So again, this is the hippocampus. You have the CA1, CA2, CA3, and intake gyrus region of the brain of the hippocampus section. And all of these are responsible for different memories and cognitive control and learning. So I took these microdissections, and this is in fact what it actually really looks like. So I took these different parts of the brain and I performed a sequencing analysis. So I apologize for a very busy slide, but what ultimately you're looking at here is our genes that are upregulated or genes that are downregulated. Those in red are up and those that are in blue are down. Ultimately, what you're looking at here is genes that are fundamentally influenced in Alzheimer's disease, and in yellow, those that are influenced by the viral infection. So what this research has uncovered here is potential inflammatory therapeutic targets for us to be able to design appropriate therapies. So hopefully in the short time I've spoken to you, I've been able to convince you that infection with a very common herpes virus is actually accelerating cognitive decline and promoting Alzheimer's disease. This infection actually increases the presence of viral-specific T cells into the brain. And you have to remember, these should not be there. So they are contributing to pathology because I've shown that via the dialytic analysis and showed that the brain function in itself is impaired upon the viral infection. And then further, interestingly, when we add these antiviral compounds, and data I haven't shown is by depleting viral-specific T cells from the system, you can actually repair and rescue any cognitive impairment. And this decreases overall inflammation, and thus you get a decrease in overall Alzheimer's disease pathology. And then finally, the sequencing analysis has allowed us to identify inflammatory genes that are being exacerbated by the virus itself, thus promoting Alzheimer's disease development. Because as I said, the viruses shouldn't be in the brain, but the viral-specific immune cells are. And then before I hand over to Viola, I just wanted to say the brain itself is protected by a blood-brain barrier. This is part of the body's own way of protecting the brain from any form, from our blood system. But ultimately, what you've seen from my research is that the viruses and the immune system associated with that viral infection is bypassing that and thus driving damage. So there's lots of people I need to thank for doing all this work, which includes my lab team here and more people here that I'd love to name individually for their contributions, but I can't. But I would just like to say none of this research would be possible without the generous funding of my funders, in particular the Hodge Foundation, the Hodge Centre for Neuropsychiatric Immunology, who funded all of the research that you saw here today. So I'll hand you over now to my esteemed colleague, Viola. So thank you very much for listening.
Thank you, Matt. So I'm also interested in neuroinflammation, and what I'm going to talk about is what we do in terms of the development of novel drugs to treat dementia. And the current dementia drugs target amyloid beta. This is the protein that is deposited in our brains. So those drugs tend to remove this aggregates with hope to improve the cognitive impairment. What those drugs do, they slow progression of the disease, but they don't cure the disease or they don't stop the disease. And the difference between the drug and the placebo in slowing down the progression is about 27%. So you can tell this is a very small difference. And people who take those drugs tend to experience severe side effects. And these include a stroke, microbleeding, and brain swelling. So for me, is a high risk for a small benefit. And we are reaching the point, as Matt was saying, that dementia becoming a global issue. And we really have to think hard how we're gonna address this and really need a safe but also effective drug to treat dementia. So Matt was talking about adaptive immune system. So I'm going to shift the focus into innate immune system. And this is the part of the immune system which is responsible for very first responses to infection. So when we cut our finger or we get COVID, the body needs to react to recover the healthy balance. And this is a good thing. Problem starts when the infection is a chronic issue and causes disease. And part of the innate immune system I'm looking for is complement system. And complement plays two roles. First is to tag the pathogens for removal of dead cells and debris. It's a waste disposal mechanism. A second part is direct killing of pathogens. So when we see on this cartoon here, we have complement protein complex called membrane complex, and Matt assembles on the surface of the bacteria cell and drills holes in it and causes death of this bacteria by lysis. So ongoing complement dysregulation leads to brain inflammation. It sets brain on fire, and we can visualize that with techniques such as PET scanning. So you can see on this picture here, lots of inflammation going on in this Alzheimer's brain compared to control, which looks very much cool. And we can actually visualize the complement pathology in this patient's brain. When we zoom on, we can see the amyloid beta plaque in green, which is surrounded by lots of complement in red. So we really know that the complement does contribute or actually drives the disease. And how complement does this is shown on the slide. So there are two mechanisms. The first one is that complement proteins activate the brain cells, such as microglia, for example, and that sets this microglia into activated angry mode and attacks on cells to kill them. A second mechanism I already introduced is a MAC attack. So MAC is a protein I'll be talking about mainly in this presentation. So MAC attacks neurons and also synapses. I'll go back to synapses later. And so many of those MACs can assemble on the surface of the neuron, causing osmotic pressure and eventually cell death and releasing the content. And those leaking cells cause lots of inflammation and disease. When we talk about anti-complement drugs, those are already being used in the clinics, mainly for renal diseases. A good example is eculizumab. It's been around for more than 15 years, and it's been used for a disease called PNH. So in this disease, we have red blood cell, which is attacked by MAC. So we have many of those drilling holes in the cell. And then eventually cell death by osmotic pressure. So when we give the drug, this prevents that mechanism to happen. And this is exactly what my antibodies do. And I say antibodies because I hope you all are familiar now with antibodies after COVID era. So these are great tools for detection, but also are very good therapeutics because they are highly specific. The question I ask is whether we can use antibodies that block MAC formation for brain diseases such as Alzheimer's. And secondly, how do we deliver those drugs into the brain? For those who work with antibodies, or you may know that antibodies are large molecules, so they don't really get into the brain. So that's why we have to think about clever mechanisms, how we can deliver those drugs into the brain. And I do gonna pause now and just apologize for not showing my primary data because of the pending patent application. So there are many ways how we can get drugs into brain. The first one is, for example, using ultrasound. So this is actually safe method and approved by FDA. So it's temporary opening of the barrier, allowing the drug to get in. Second one is direct delivery. You can imagine this method is quite invasive and also not feasible when we think about using it for a common disease such as Alzheimer's, where we need a frequent dosing. Third one is to design a perfect drug. So it gets into the brain, and this is something we are working on, and usually is related to the size of the drug. And then we can use cargo delivery, so encapsulating the antibody into a vehicle to deliver to the brain. And finally, we can use shuttles, and this is something I will be talking about here. So this is using Trojan-horse delivery system. So by using shuttles, I mean tagging the antibody to that shuttle, and the shuttle allows smuggling that antibody into the brain in its active form and release. And I have made those proteins in a large quantity and expressed them, tested in tube tests and in brain to see whether there's impact on the disease progression, which I will be showing you in the next slides. So in a tube test, to look for a brain penetrance, I set up the system where we have a well where we grow the antibodies, in the cells as a monolayer, and this creates this barrier. And so this is the blood site. And then we can add the drug, and we can measure very precisely how much of the drug goes across this artificial barrier to the brain site, and can plot that data, and we can see that the shuttle is much more efficient in doing so compared to control, which is just irrelevant fused protein. But of course, more importantly, we wanted to know whether there's any impact on the Alzheimer's disease. So for this, we use the same method Matt introduced. And when we zoom on the connections between the neurons here, you can see those synapses which communicate, exchanging information, neurotransmitters, proteins, and so on. And then under microscopes, we're looking for those shapes and then the buttons around here. So the more of those there is more communication going on, the better for the brain. And hopefully what you can see, there is much more of those formed here in the drug-treated brains compared to controls. So this is a very exciting finding, only 1 week of treatment, and we are following out now with a longer one. So what we have is a proof of concept that we can use anti-complement drugs for brain diseases such as Alzheimer. We can deliver them to the brain in a safe mode, and they show to be effective in neurodegeneration. And we are very keen to improve those drugs to improve their strength of binding and ability to penetrate into the brain. And of course, it would be great to see them moving into human clinical trials. So with this, I'd like to say thank you to everyone who was involved with this project, and all the collaborators, and Professor Morgan, and my mentor, and my team, and of course, Race Against Dementia, Formula One, my founder, because none of this would happen without their support. So thank you very much.
Well, thank you very much indeed, both of you. That was absolutely fascinating. So essentially, I think what you've been telling us is that we've been barking up, not exactly barking up the wrong tree, but we've been focusing our research for years on the amyloid B plaques, these deposits of proteins in the brains, and not really getting us very far. But what you're looking at is what's, we now understand it's inflammation of the brain that's the problem, and let's target that, which is what your work is doing. And we saw some results there, Wiola, which were really interesting, and let's hope that then develops. So what I'll do is, we had a couple of questions in. That's great, thanks very much. I'm just going to start with some pre-submitted questions. I think some of them could be answered fairly quickly. I've got a few of them, so if you want to be relatively brief in answering these. Well, the first one's quite a big one, actually. In what way are our constantly accelerating lifestyles affecting our probability of developing dementia in the future? So I take that to mean things like stress and diet and alcohol, lack of exercise, that sort of thing.
Do you want to take it? Yeah. I'll take it. So yeah, it's really well known that stress induces the immune system to be activated. So too much stress is going to result in too much immune activation and could potentially contribute to developing more forms of dementia, vascular dementia, etc. Also, Alzheimer's disease is associated with lifestyle, socioeconomic status, so smoking, drinking, obesity. So yeah, all of these things are definitely linked. So I wouldn't, I'm not a clinician, but we do know that stress induces the immune system. So if you were to ask me, I would say that as best you can, try not to be as stressed as one can be.
And if I can just add on to it, if we're talking about accelerated lifestyle, I think being busy is actually good for us. Keeps our brains switch on, and it's actually, there are proofs that the people in the higher education sector are of less risk of developing dementia.
So keep your brain active, but have a good lifestyle. We all know roughly what that means. Yeah. So there's another related one. What affects brain inflammation? Do food intolerances have an effect on brain inflammation?
I wouldn't say it's intolerances. There is a lot of data that suggests the microbiome or the gut flora that lives in one's gut, and a healthy gut is associated with less Alzheimer's disease onset. There's actually work from Cardiff that's recently published that shows that. I'm not sure in the context of intolerances to specific foods. I'm not sure that data exists, but definitely a healthy gut microbiota is one that would prevent Alzheimer's disease progression. As I said, that's definitely, that's been critically reviewed and published by researchers here at Cardiff.
Excellent. Eat plenty of plants.
Yes.
Get that microbiome. Yeah. Another one. Do immunosuppressive treatments like those used to treat myeloma influence the onset of or clinical course of dementia. So it's about immunosuppressive treatments.
So I think it's not really clear yet from the literature. And problem with those drugs I have is that we don't know, or they don't really get into the brain and they're not specific. So even they might not, but I think we still have to actually do a proper research studies to investigate that.
Yeah, I completely agree with you. I think, I'm not really sure that data exists to confirm or deny anything there associated with other immunosuppressives. It depends what you read in the literature. Some people have written that too much immunosuppression is bad for developing Alzheimer's disease, whereas other papers have shown that it's the converse. So, you're trying to weigh up a very fine balance here between tipping the system one way or another.
Yeah, so that's really not conclusive, and probably nothing to worry about in that sense. Another one here, interesting one. Can stimulating blood flow at the nape of the neck help?
I wouldn't have thought so. Personally, I wouldn't have thought so, based on the fact that, that's obviously a very sensitive area. So blood flow into the brain is fundamentally vital. But obviously what both Wiola and I have just talked about, that I don't think it's stopping the blood getting into the brain, it's stopping the wrong things that exist within the blood into the brain, be that a virus, a viral infection, or an inflammatory response.
I agree. I think we don't really know, but I could say that I would be more worried about conditions of the veins. We know that vascular dementia is the second biggest cause of dementia. Focusing on that.
There's nothing to suggest that might help, no. And a final pre-submitted one, then I'll come to the ones that you've been very helpfully typing in. Are there warning signs at the age of 45 to 50? Is forgetfulness in a busy schedule a sign?
Shall I go first?
Yeah,
so that's an interesting question, based on the fact that there are known genetic risk factors for developing Alzheimer's disease, and at 45 to 50, that would be more of the early onset AD, and that's associated with your genetics, so you would probably be aware. And that's much more rare. It's only about 5% of Alzheimer's disease cases. However, with all of if as my second slide, I showed all of the associated loss, so concentration, mood, physiospatial awareness. If you have a combination of those events, then I think it would be time to see your GP. But if it's just one, you could put it down to anything, just a busy lifestyle, or just you feeling tired that day, but yeah, I think it would be a combination of multiple events that one would need to be more worried.
Yes, I agree. It's a complex disease presented with an umbrella of symptoms, not just the one symptom. So forgetfulness, actually one of the a normal part of the normal aging. Worry about it. But I'm not a clinician, so.
I'm just being very busy, as you said, as the questioner implies. So next question that's come in. Well, what's in the pipeline? What's the next stage of your research in investigating this problem?
For me, it would be moving the drug towards the clinical trials. So this is what I'm working hard on now, and I'm trying to find extra funding to support this work, and actually set up a bigger study to confirm the preliminary data, and yes, test in a small subset of people to see whether there is any effectiveness in a cognitive impairment.
And for me, it would be, again, similar to Wiola, sourcing money to do a larger body of work. I've shown the preliminary data that shows that the virus does, in fact, indeed exacerbate Alzheimer's disease, but we need to do this much larger, much broader level of research. You can do this in humans and find immune-mediated regulatory prognostic markers. So using the immune system as a way of identifying Alzheimer's disease onset. So that would be my long-term goal.
Because it'd be a real advance if we could identify it at a much earlier stage, wouldn't it?
Okay, that's right.
No reliance on symptoms actually come in at quite late stage. The next question is about the use of data science. Is there a role for machine learning algorithms in your research?
It's an interesting question. I mean, when we look at big data, probably something that Wiola and I go to is already published data sets. So similarly to the sequencing analysis I showed you of that large heat map. So there's lots of data in the literature that's already published that is freely and publicly available that both Wiola and I will actively use to compare our data set against, because that's the right thing to do. Machine learning is an interesting question because systems immunity or systems analysis is becoming much more prevalent in our research. So we haven't actually done any machine learning, but we do use bioinformaticians and statistician approaches to compare all of our large sequencing and GWAS data sets that are freely available that we compare our data against. Is that true, do you think?
Yeah, I think that's well explained. We recently completed a study where we look for proteins levels in the blood of Alzheimer's patients, and we linked this back into a GWAS studies of the genetics. And we found that novel genes which are responsible, or might be responsible for the pathology. So this is an incredible tool and it's very helpful, and we are leveraging into those resources, and we are really lucky to have them in Cardiff. As you said at the beginning, we are a very unique, it's only 2 places in the world we can do that kind of research. So that's fantastic.
Yes, absolutely wonderful. And then there's one coming back to your research, Matt, I think, and somebody asking whether antivirals might help ward off dementia. And I think you sort of partially answered that, but what's your response?
Yeah, it's really interesting because there's lots of viral infections and some severe chronic infections such as flu and HIV that causes mass scale delirium. And antivirals are known to instantaneously remove that delirium, which is another form of cognitive ability in those patients. So I don't think long-term an individual should be on antivirals because some of them can be quite toxic. However, the new ones that have been developed because of COVID are much more well tolerated. So I think if we have a way of combining a prognostic marker of those who are at risk of developing Alzheimer's disease, and then incorporating that with a mild, just ticking over antiviral in the background for those patients, I really strongly believe that you're going to delay the onset of their Alzheimer's. So yes, I think they have an important role to play moving forward.
Interesting. There's a related one now. Are there other viral infections indicated in the onset of dementia that you'd like to extend your research into?
Yes, I would. It's known in Alzheimer's disease that there are other human herpes viruses that are found in the brain of Alzheimer's disease patients. So there's human herpes virus 6A and 7. But there's lots of viruses, and I don't think it's the virus per se itself that's actually driving the disease, because it's difficult to find the virus itself in the brain. My research as a T-cell viral immunologist focuses on the fact that it's the T cells that are driving the disease. It's the immune system that's been hyperactivated because of the infection that's driving your immune system to become hyperactive, rightly so, to remove the virus. And then what's happening is you're having a byproduct of that inflammation that's then contributing to the infection, because it gets into the brain and then causes this damage. So any chronic infection that a human may have that elicits such a strong response, and indeed CMV elicits one of the strongest ones known to man, is going to possibly contribute to this disease. So as I said, this is true in HIV and also flu.
Somebody has asked about COVID in the questions, where COVID affecting the brain. Is that a...
Yeah, COVID, yeah, it's a good question. COVID is found in the brain of people who have sadly died of COVID. The link between COVID and Alzheimer's disease is quite tenuous as we speak, obviously, because it's very early days. The fact of the matter is that lots of, sadly, Alzheimer's disease patients died in the very first wave of COVID, but that was more opportunistic as to they were living in care homes, and the virus is able to go through these places quite rapidly, untouched. So long term, I believe that research will come out, and there is data that suggests that some of the brain resident cells would allow COVID to get into them. So you get viral entry into the brain resident cells. So that's true with any coronavirus, not just SARS-CoV-2. They're called neurotropic viruses. So they're known to live quite happily within the brain, but most of the time they don't cause any problem. However, with SARS-CoV-2 and Alzheimer's disease, that's a very tenuous link right now, just because it's very early days.
So this is really promising research, and obviously there's lots of research, a huge amount, well, not enough probably, but there is research going on in this area. Can we hope one day to be able to actually prevent and eradicate and cure Alzheimer's, or prevent it, or both, and other forms of dementia perhaps?
Yeah, I think we should be very optimistic about the future. We checked what does not work and we know what might work. So I think we've spent last good couple of decades studying amyloid, and we know that it's a very good biomarker for the disease, but not necessarily at the cause of the disease. So we do have to explore other pathways, and inflammation is a big part of Alzheimer's pathology. And as thought before, it could be the outcome, of the disease. But now we know that it's more likely that it actually is the cause of the disease, and this is what we are investigating and trying to confirm.
Yeah, I completely agree. And I think this is where Cardiff is very unique, because we have world expertise in both neuropsychiatric techniques and also infection and immunity. So as Wiola and I have said, we truly believe that it's an inflammatory mediated disease. So by merging these two research disciplines, Cardiff is in a really unique position to lead on this research. And ultimately, I do believe that we will find a cure or a prognostic marker that's associated with the immune system. Hopefully, as soon as we can.
That certainly sounds very promising. Maybe you've got time for one or two last questions before we finish at 6. A couple of people have asked whether this approach is applicable in other diseases as well. Somebody mentioned cancer.
Yes, definitely. I think you probably mean the drugs, definitely.
They are anti-inflammatory. They stop at the very vicious cycle of enhancing inflammation. So we actually, as I said, some of them are already being used for kidney diseases to stop the MAC formation, and MAC is the most pro-inflammatory product of the complement cascade. And they've been tested in other clinical trials, which are still ongoing. There's several in the pipelines for different diseases, such as Huntington disease, ALS, and other kidney diseases. So yes, there's quite a few, and eye disease is a big one. So AMD is really promising, moving forward rapidly into FDA approval.
Okay, well, it really does sound as though you're investigating some very promising lines here, which are really getting into the mechanisms and actually affecting the progress of this disease. It was very striking, your slide over there on the neurons, because in the end it comes down to that. If we can restore those, that's what we need, as long as they then function normally. So I think we are pretty much out of time now. It's a time to draw this to a close. So thank you both very much indeed for your talks and answering the questions. And also thank you very much to you, our webinar audience, for being with us. And I hope you found it as interesting as I did. And thank you for the questions. If we didn't get a chance to answer your questions on screen now, we will follow up with you directly. So we'll ask Matt and Wiola to answer the question, and then we'll get in touch and let you know what that was. All of you will receive an email in the coming days with a copy of the recording and links to register for future events. We hold these events regularly, so do look out for them. The next event in the series is taking place on Thursday, the 20th of April. And there we're going to hear about the research taking place here to understand how Cardiff University's research community responded to the COVID pandemic and how we're contributing to preparations for any future pandemics. And I'm sure that's going to be a really fascinating session, because we did actually do an awful lot in that respect. If anyone watching would like to get involved with Cardiff University, volunteering time to help our students or helping us to fundraise for our research, please do follow the link in the comments box, which is there for you now. That's it. Thanks very much, everybody. Once again, goodbye. Diolch, hwyl fawr.